I think I have been speaking enough in the previous lecture, so I thought we would go essentially over the same thing, but reading a paper together. This is one of the latest review paper written by a very famous guy in the field, although nowadays he just publishes reviews every year instead of doing actually research. But I mean, he deserves it, I suppose. So I thought we might actually go together over the paper. It won't be so much benefit for you because I know you can read better than I do, but we could maybe, each of us could actually read one paragraph, and then if there is anything you do not understand, then you can ask questions, and it's a little bit more like a discussion. And if there is anything, don't hesitate. It can be vocabulary, or it can be biological knowledge, or anything. If there is anything you don't understand, ask a question. Socio? Ne? So please, start.
Transcriptional architecture of the mammalian circadian clock. Abstract. Circadian clocks are endogenous oscillators that control 24-hour physiological and behavioral processes in organisms. These cell-autonomous clocks are composed of a transcription-translation-based autoregulatory feedback loop. With the development of next-generation sequencing approaches, biochemical and genomic insights into circadian function have recently come into focus. Genome-wide analysis of the clock transcriptional feedback loop have revealed a global circadian regulation of processes such as transcription factor occupancy, RNA polymerase II recruitment and initiation, nascent transcription, and chromatin remodeling. The genomic targets of circadian clock are pervasive and are intimately linked to the regulation of metabolism, cell growth, and physiology.
Very good. It's only the abstract, so there might be words you are unfamiliar with it, and they may get explained later. But so we are going to read this thing together, and so far only the first abstract has been read. So, is there any word would you like me to explain from the abstract? There are probably, I can imagine there are a few.
Nascent transcription.
Okay, that's a very important one. So it's nascent, it's relatively new. Maybe over the last 5 years this became very big because of the invention of new techniques to measure gene expression. So you all know the well-known central dogma in which you have DNA is being transcribed into RNA, and then you have the RNA with the poly A tail, and then the RNA goes into the cytoplasm, this is the nucleus, goes into the cytoplasm and is translated into protein, right? This is protein. And usually it's not exactly true, but it's close enough. Usually what people measure is the amount of mature RNA. The amount of RNA that you have at a given time. For example, say 12 hours after the beginning of the day, you measure how much RNA you have in your cells. One specific RNA. So let's say, for example, RNA from gene number 1. You measure that RNA. But actually what you really don't know when you measure how much of a mature RNA you have is how much RNA is made and how much RNA is degraded. You don't know the rate of synthesis of that RNA. You know how much there is, but you don't know how quickly it is degraded and how quickly it is made. So it is not dynamic. So what people are now doing is that instead of measuring the amount of this this is English is called steady state. Steady state. So for example, steady state, imagine you have a box of apple and you have in the box 20 apples. But in the morning you have the shopkeeper, he is putting apple into the box, and when it's filled there are 20 apples. But during the day people come and buy apple. So at the end of the day in the box you have no more apple. So it means that the turnover is every day 20 apples are added, and at the end of the day 20 new apples are added. So if in the morning if you measure the amount of apple, you say okay there are 20 apples, and in the evening there may not be any more apple. But actually you don't know how many new apples are created and how many apples are being taken. So this is the same kind of thing. So this is the steady state amount of apple, maybe 20 for example. So what people are doing with RNA is that they actually measure how often one RNA is made. What is the rate of the synthesis of RNA? How many copies of this RNA are made at every single moment? It's a little bit more dynamic. What RNAs are being synthesized at one given time? This is a little, do you understand the difference? It's not how much RNA has been made, it's how much RNA is being made at that time. This is what is called nascent seq. And in English, nascent, it means being made, being synthesized. This is what is called nascent. What else? What other words in the abstract? We need some clarification. Next generation sequencing approach. You understand?
No.
Okay. Do you understand next?
So it's like the future.
Yeah, so sequencing. Many years ago, a guy called Sanger, it's an inventor, he invented a method to sequence DNA. So you take a bit of DNA, you extract DNA from a cell, and you can actually read what is the sequence of that DNA. But then the next generation sequence was invented. It's actually not so long ago. And what this enables you to do is not only sequence a mix of different bits of DNA, but it's also quantify how much copies of each sequence you have. Not only sequence, and by the sequence you know the identity of what you have, but it also measures how many copies of fragments of the same sequence you have. And not only you can do that with DNA, but you can also do that with RNA. So imagine you extract RNA from a cell, and with this next generation sequencing approach, you know what is the sequence of this messenger RNA and how much of it you have. So then it becomes very dynamic, and you can measure essentially whatever the cell does at the RNA level. So that's very simple. Is that okay? Okay. What else? Is it okay? RNA polymerase II recruitment and initiation. Is that fine?
Standard transcription.
Yes.
Beginning.
Yes, exactly. So RNA polymerase. So in cells there are essentially 3 RNA polymerase, and the RNA polymerase II is the protein that's or the complex of protein that synthesize messenger RNA. This is the RNA polymerase II, and it is recruited in the promoter of certain genes to initiate the transcription. chromatin remodeling. This might be also a little bit new. Chromatin remodeling. So the DNA in a cell, the DNA in the cell is not new. So in your cell, you don't have a big noodle that looks like that is DNA. This is not at all like that. If I draw it as a kind of cartoon, so imagine this is DNA, right? And I don't show, but this is of course a double-stranded bit of DNA, right? Maybe I can show a small fraction like this. Right? This is your double helix of DNA. In the cell, it is not like this. Actually, this bit of DNA, the genomic DNA, is coiled around protein that are called histones, and they are it's a complex of I think 8 different types of proteins, but the DNA is wound around these histones like this, and it's packed up around protein. Okay, but then you can imagine, what if I want to transcribe the gene that is there between two proteins? It's kind of difficult. So, for that, you must actually, I will draw this again. If I want to transcribe the gene that is there, I need to remodel this. And these proteins, they are called histones. So the whole thing is called the chromatin. And then what I will do, I may actually unroll this one here so that the DNA here is a little bit more relaxed. And then I can have RNAs, polymerase II, that can access this gene and transcribe it. And this is called the remodeling of chromatin. So it's very dynamic.
Okay. Okay.
Okay. All right. Let's go for the next paragraph, from energetic to debate, and that will be, I don't remember what is your name. Ifan, please.
I read it or?
You read, please.
Energetic cycles governed by the solar day have driven the evolution of life on Earth. To adapt and respond optimally to the daily environmental cycles, living systems utilize internal timing systems that resonate with the 24-hour day. Across life forms, the internal timing system can be seen at the transcriptional level, giving rise to divergent gene networks in different organisms that oscillate on a 24-hour basis. Circadian clocks, as these endogenous timers are known, seem to be the product of convergent evolution, as the design principles of circadian transcriptional circuits share a common network motif, that is, negative feedback with delay. Although the specific components are divergent across the kingdoms of life, the link to the daily solar energetic cycle is most obvious in photosynthetic organisms, but is also a conserved motif in the neutron cycles of higher organisms. Whether circadian clocks evolved to adapt to the energetic demands of the environment or to avoid the detrimental consequences of solar radiation remains a topic of debate.
Okay, thank you very much. So that's a very nice paragraph that essentially synthesizes many of the things we have seen so far. So, and this is of course associated to the figure 1, and the figure 1 is here. I can make it a little bit smaller. So, of course, there is the rotation of the Earth with the light and dark, with the 24-hour cycle, and the behavioral rhythms mirror this rotation of the Earth. So we have kind of encoded this one into our biology, and the most obvious rhythm is really the wake-sleep rhythm. But the wake-sleep rhythm also means feeding-fasting rhythm. We only feed during the active phase. And then that we have seen before, right? So you have some responsive element in the DNA, part of the promoter of clock genes, and these are activated, and the clock genes when they get transcribed and translated, they form negative elements and inhibit their own production. So that's a very, very simplified way of how the clock works. And you may remember that these two proteins may represent clock and BMAL, which is the activator, and this clock gene may be, for example, PER gene, which when it is made inhibits its own transcription by inhibiting clock and BMAL. And then what we have seen before as well is that if you actually measure rhythms in different cells in the body, then not only you can see nice rhythms in the SCN, in vivo, so in vitro it means in a dish, or in vivo, if you actually really measure in an organism that is still alive, you have these beautiful oscillations of clock gene expression. But you can measure the same thing in the lung, in fibroblasts. It's kind of essentially skin cells or whatever cells. Fibroblast is kind of a cells that is present in probably all the body and make up the bulk of our tissue. And then you have also the same thing in the liver. So you have these rhythms everywhere. Okay. Any questions about this paragraph? Don't hesitate. There are no stupid questions, only stupid answers.
What is convergent evolution?
That's a good one. So I believe these words, this is a very good review. These words that are written in the different characters, you have the definition next. So convergent evolution is the process by which unrelated organisms independently evolve similar traits as a result of adapting to similar environments or selective pressure. Do you look at me like it's written here?
Oh.
You see, because it's written in a different character, the definition is given there.
Oh.
So this is why I chose this review, because it's very, very nice. So convergent evolution means essentially two different types of organisms. They essentially invent the same thing. So two people, one in Italy, one in the United States, they both invent the telephone. But only the one from the United States makes a patent. So he becomes the inventor in history. Anyway, it doesn't matter. But yeah, convergent evolution is the same thing is evolved at the same time but independently due to similar pressures. What else? Network motif is also defined next to the text. Any other thing in this should be okay, ne? What about kingdoms of life? You understand kingdoms of life? So this is another the evolution tree, right? The kingdoms of life. What are the kingdoms of life? What are the kingdoms of life?
7.
Which are?
Oh God.
What are the kingdoms of life? Who knows what are? It's changing. It has been changing almost every decade or every 5 years. What are the kingdoms of life? The archaea is one.
I thought that they're domains, aren't they? Of Archaeobacteria and Animalia.
No, they are not 3. They are more than that. They are.
There's 7.
I think
There's like
plants, Animalia, Plantae, Archaea, fungi, protists. So they are the major families of living things. And actually there are several different classifications even of these things. But essentially it's very difficult to go higher than that. Then the rest is just life and not living. So this is the same thing. Okay, what is it okay? So we can maybe go to the next paragraph. Please, Vincent.
Irrespective of why circadian clocks evolve, it is evident today that cell-autonomous clocks are ubiquitous. In mammals, for example, the neurocentric view of a master clock located only in the brain is no longer viable. Instead, the discovery of clock genes in the 1990s showed that virtually all cells express these genes and have the capacity to generate circadian oscillations. The circadian clock in mammals is now conceptualized as a hierarchical system in which a clock located in the hypothalamic suprachiasmatic nucleus acts as a master pacemaker to synchronize or entrain peripheral clocks distributed throughout the body. This system-wide architecture raises the following question. How is this hierarchy coupled and integrated? And what are the functions of clocks in tissue-specific and cell-specific contexts?
Thank you very much. What about this one? Oscillations is defined on the side. Any other ubiquitous is just English. Do you understand ubiquitous?
Everywhere.
Yes.
Hypothalamic suprachiasmatic nucleus.
これは視床下部の
視床下部
視
の
交座上核。視交座上核。視交座。You want to see kanji? You want to see kanji? You want to see kanji? Suprachiasmatic nucleus. Yeah, I mentioned it in previous lecture, but if I only say it, it's difficult. So, 視交座上核 isちょっと待ってね。It may not be at all. I will not even try to write in Japanese, but I'm trying to find the definition.
Okay, I will try it.
Nucleus, and then Japanese. Shikoza Jokaku. Shikoza. I will just get the. Yeah, Wikipedia. You know, I'm a very, very good teacher. You know, using Wikipedia as my source. That's it. That's the one here. This is correct. Okay, this is correct. I would not show you any rubbish from Wikipedia. This is correct. Now you have a lot of rubbish on Wikipedia. It's you have always to be careful. So yes, so actually it just means the nucleus that is over the optic chiasm. And in Japanese, this is also what it means. You know where it is, right?
Nerve. Is it nerve cell is crossing?
So where it is that in human you have in mouse is similar. You have the brain in human, and here this is the cerebellum, and then here you have the shishokabu, the hypothalamus. Then you have the eyes here, right? It's not necessary, but then you have the optic nerves. They kind of converge at one specific point, and then they cross each other. Information from the left eye goes to the right brain, and from the right eye goes to the left brain. And when they cross each other, this is called the optic chiasm. The optic chiasm. And then over the optic chiasm, you have the suprachiasmatic nucleus. It's in the hypothalamus. This is what it means. It's okay, you understand this one. Okay. What else? Is it okay? Okay. You can ask questions at any time, even if it's about something we have already read. Okay, please read the next one.
Okay. The molecular mechanism of the circadian clock in mammals is generated by a cell-autonomous transcriptional autoregulatory feedback loop. The core clock genes include clock and BMAL1, which encode activators, and PER1, PER2, CRY1, and CRY2, which encode repressors. Current topics of active investigation include the components of this core circadian clock gene network, how environmental and systemic inputs impinge on the core clock mechanism, and how the core clock network regulates downstream output pathways. Recent work has provided insight into the genomic targets of the core clock pathway and has led to some surprises concerning the pervasiveness of circadian regulation in gene expression. A substantial fraction, 5 to 20% of genes expressed in any particular cell or tissue have been found to undergo circadian oscillations at the mRNA level. However, this level of regulation, that is, steady-state mRNA rhythms, is only one of a multitude of layers of circadian regulation in gene expression. Virtually every regulatory stage in gene expression that has been examined in detail, including transcription, splicing, termination, polyadenylation, nuclear export, microRNA regulation, translation, and RNA degradation, has revealed additional layers of circadian control. Thus, both transcriptional and post-transcriptional regulation of circadian gene expression are key nodal points of control.
Okay, thank you very much. So essentially this contains most of what we have seen over the past lectures, but in addition, what we saw so far mostly was about the transcription and how the transcription of gene is regulated by clock BMAL, CRY1, CRY2, PER1, PER2. But what it says here is that actually it's not only really the transcription, that's only one layer of the thing, but there are other many things like RNA processing, translation, degradation. They are also regulated, and they are also layers of the circadian clock. So seeing the circadian clock as only a transcription feedback loop may actually not be very correct. It is only one part of the thing, but it may be the most important part, because if you disturb, you have no more rhythm, but other layers are needed for it to function correctly. Okay? Say, for example, if you inhibit RNA degradation, or if you decrease the rate of RNA degradation, what do you think might happen? It's a very easy thing. Imagine you decrease the rate at which the RNA of clock gene is degraded. Per 2 mRNA, for example.
Will it slow down the rhythm?
It will slow down the rhythm, and the period of the clock may become longer, 30 hours or more. Okay? Any questions about this paragraph? It was quite long. Any vocabulary or concepts?
When it says undergo circadian oscillations at the mRNA level, does that just mean that that's where they observe the oscillations in the mRNA?
Yes, so it means that, let's say you have a lot of mice and you extract whatever tissue, say the liver, every 2 hours, and you measure specific mRNA at different times every 2 hours, then you see that the amount of mRNA actually oscillates. Yes.
So it's kind of like a static point.
It's like a static point. But if you measure gene expression, yeah, so essentially you measure RNA. You measure the amount of RNA, or the amount of protein also can be measured like that. But we will see that later, but you can also measure more dynamically by using some nice technique, which I will show you later. What about all these transcription, splicing, termination, polyadenylation, nuclear export, microRNA regulation, translation, and RNA degradation. Anything that requires explanation? That's okay? No? What about the vocabulary, like what is pervasive? Pervasiveness. That's not so common word. Pervasive means it's a little bit like ubiquitous. It's just everything, everywhere. It can be found everywhere. This is very similar. What about? I saw one maybe. Yeah, cell autonomous. That's fine. Yeah. Okay. So anyway, we can always go back if you have questions. So I will read the next thing. Our understanding of the mechanism of the circadian clock in mammals has been informed by progress on two fronts. First, by the biochemical definition of components of circadian activator and repressor complexes, and second, by the structural determination of key domains of clock protein, which is box 1, we will see later. This review focuses on the molecular, biochemical, and genomic advances in our understanding of circadian regulation in mammals. I highlight work on the genomic targets of the core circadian transcriptional pathway, as well as studies revealing global circadian oscillations in RNA polymerase II mediated transcription and their relation to the dynamic circadian regulation of chromatin state and chromatin modifiers. For non-mammalian model systems and aspects of circadian biology focused on photic regulation, metabolism, and immunity, there are other excellent reviews. It is not written here, but what I mostly review in this paper is mostly what my own team has done. He has not written that, but that's what he has done. Okay, so the thing that he reviews here essentially is his own work. So it's by no means complete. Okay, this guy mainly reviews his own work and the work of his minions. Any question? That should be fine.
Photic regulation.
どこだった？
Last.
Photic regulation. なんだろうね、photic regulation. What does it look like? Photic regulation. What does it look like? The word photic.
Photon.
Photon. Yes. What is a photon?
Light particle.
Yeah. So this is actually the regulation of clock by light.
Oh.
Not the endogenous mechanism that allows the cell to keep on oscillating, but how the clock entrains the biological clock. This is the photic regulation. Okay. And next. Box one. We need to see box one. Sorry. I'm interrupting your stars there. Where is the box one? Yeah, so that's the box one there. But you can look. I will not go into the detail here. You can look at it by yourself. But this guy, Joe Takahashi, he has done a lot of work about really the structure of the protein, and he has really shown how the different proteins, for example, A here, I don't even know which this is. It might be FBXL3 or something. So he has a lot of work about the structure of all these proteins. Where do they bind to? How do they bind? What amino acids they are composed of? What is their active site? So, for example, here this is the PER protein with the phosphorylation domain and everything. So let's not go into detail. I don't think this is necessary, but for those who are interested, it's kind of interesting. And studying this enables us really to understand how it works and maybe even make like medicines and things like this that can target specific sites of these proteins. So it is interesting. But please read this one by yourself. Okay. We will just go over the main text, please.
Okay. The core circadian clock gene network. In mammals, the mechanism of the circadian clock consists of the activator's clock and its paralog MPAS2 and its heterodimeric partner BMAL1, also known as ARNTL, which are basic helix-loop-helix transcription factors. The clock BMAL complex binds to regulatory elements containing E-boxes in a set of rhythmic genes that encode the repressor proteins period, encoded by PER1, PER2 and PER3, and cryptochrome, encoded by CRY1 and CRY2. In mice, clock BMAL activation occurs in the daytime, leading to the transcription of the PER and CRY genes in the afternoon, and the accumulation of the PER and CRY proteins in the late afternoon or evening. The PER and CRY proteins interact with each other as well as with the serine threonine kinases, casein kinase 1 delta, and CK1
Epsilon.
epsilon level, and translocate into the nucleus at night, where they interact with clock BMAL to repress their own transcription. As repression progresses, PER and CRY transcription declines, and PER and CRY protein levels decrease. The half-lives of PER and CRY proteins are relatively short and specific E3 ligase complexes target these proteins for ubiquitination and subsequent degradation by the proteasome. Once negative feedback repression is relieved by turnover of the repressor complex, transcription by clock BMAL1 can begin anew to start a new cycle of transcription in the next morning. Importantly, core clock genes, such as those that encode PER2 and its regulatory kinase, CK1 delta, have been found to underlie human sleep timing disorders, thus demonstrating a clear role for clock genes in humans.
Okay, thank you very much. Okay, so essentially this is what we have seen in the last few lectures, is that you have the basic oscillatory loop, but translation and all these things are also regulated, so that not only you have cycles of RNA rhythms, but then slightly delayed, you have the cycles of the protein rhythms. So do you have any questions? We also saw the casein kinase 1 delta and 1 epsilon, which essentially phosphorylate these proteins. And everything is shown in this figure here, which is something very similar to what we have seen before. Essentially, you have the BMAL1 here, and BMAL1 and clock, maybe clock is somewhere, no, clock is not shown as a gene here, but essentially you have BMAL clock that activates the transcription of many genes which have an E-box in their promoter, and then all these PER genes and CRY genes, they inhibit this E-box. But on the other hand, you have genes like DBP and MFIL3, they sort of constitute another loop that also regulates their own output. And you also have the ROR alpha and ROR beta and reverb that can also constitute yet another loop. We saw already this one here. You can take a look at this at your leisure. So that's just the feedback loop. But then you have also kinases that phosphorylate some of these components and either controls where do they go into the cell or how quickly they degrade. Okay? And when they mention this ligase, it means ubiquitin ligase. So these are proteins which will add ubiquitin peptides to different proteins and say, please degrade this protein very quickly. For example, beta-TLCP is one of these ubiquitin ligase that will add ubiquitin to PER and leads to its degradation. Ubiquitin, this is shown here as a UB. For example, FBXL21 and also FBXL3 exist. They degrade CRY1 in pretty much the same way as beta-TLCP degrades PER. So there are many, many different layers. The question I have to you is, why so many interlocked feedback loops? You could make a clock that is only composed of, let's say, clock, beamer, purr, and cry. Clock and beamer activates the expression of purr, and when purr goes high, it starts to inhibit its own transcription. Why do we need so many different things? Of course, I told you that many different types of clock control genes have their own phase of expression, right? So having different loops, you can then have genes that are highly expressed at night or genes that are highly expressed during the day. So that's one of the reasons. But if you don't think about the output of the clock, if you only think about the clock itself, what might be one advantage of having so many loops that are co-regulating each other?
If one thing goes wrong, the entire clock doesn't break.
Yeah, if one thing goes wrong, the other clock doesn't break. How would you say that in a different way?
Checkpoints?
Checkpoints?
We need redundant mechanisms so as not because it is important. So we need redundant mechanism.
Redundancy? It's redundancy, but redundancy it's a little bit. Yeah. Some parts are not needed unless something breaks. But actually, many things may be needed for the clock to function correctly. So how would you say this? How would you say this? What do these different loops confer to the clock? What kind of characteristics is conferred to the clock by these interlocking loops? Which essentially is a word that means what you said.
Stability?
Stability, yeah, that's one. Stability. It confers stability to the system. What other words could you use? This is a word that is often used in this kind of circadian literature.
Robustness.
Yes, robustness. Stability and robustness. So all these loops, not only they will define different time frame of output, but they will also confer robustness to the system. All right. And next paragraph. Please,
sorry.
we keep the same.
Sorry,
Oh, yes, you. Sorry. Yes.
Cryptochrome.
Yeah. Cryptochrome is the full name of cry. Why it is called cryptochrome? That's an interesting question. Very often I have not said the full name of the genes. Per is very easy. It's period. Because the first time that a per gene was discovered, it's because it affected the period of the clock. But cryptochrome is an interesting word. Cryptochrome, what does it mean it's always nice to look at etymology of words because it has very idea. So it's made of two words, crypto and chrome.
Encryption.
Encryption, yeah. It's similar. So chrome is usually reserved for something that has something to do with light, like phytochrome, for example, or something that can receive light or has some relationship to light. And this chrome came because cryptochrome has the structure of the protein is very similar to a protein called photolyase, and it's an enzyme that can repair DNA activated by light. So this is the chrome part. So in the cryptochrome protein, there is a cofactor which is not made of protein, but that is something that is usually in proteins to receive light and be activated by light. But crypto it's because nobody knows its function. Crypto means wakaranai. So it's something that receives light, but nobody knows its function. So that was the first name of the thing. So this is why it's called cryptochrome. And we know now that in mammals, cryptochrome does not receive light. Maybe it originates from a protein that can repair DNA when activated by light, but in mammals, it evolved into a transcription factor that can still bind DNA, but has no relation to light. Also, in Drosophila, cryptochrome can receive light. So it's a little bit different. In mammals, it's a little bit different. What else? Okay? Alright, then let's continue.
In addition to the per and pri-target genes, the clock DMAL1 complex also activates the nuclear receptors REB, URB alpha, and REB, URB beta, encoded by NR1D1 and NR1D2 respectively, which compete at REB-DR2 and retinoic acid-related orphan receptor binding elements RORES with ROR alpha, ROR beta, and ROR gamma.
That's so boring to read.
Included by RORA, RORB, and RORC respectively. The rhythmic expression of REB-URB alpha and REB-URB beta leads to the repression of BMAL1 and clock, which induces a rhythm in these genes that is in antiphase with per expression rhythm. These interlocked feedback loops of activators and repressors form a canonical positive feedback and negative feedback gene network motif. A third clock DMAL1 driven transcriptional loop involves the PAR-B-zip proline and acidic amino acid rich basic leucine zipper, factors DBP, D-box binding protein, TEF, thyrotroph, embryonic factor, and HLF, hepatic leukemia factor. These proteins interact as sites containing D boxes with the repressor NFIL3, nuclear factor interleukin-3 regulated, also known as E4BP4, which is driven by the REB-URB aurora loop. Together, these three interlocking transcriptional feedback loops can generate cycles of transcription with various phases of expression depending on the combination of cis elements, that is, E boxes, RREs, and D boxes in the promoters and enhancers of specific target genes.
Thank you very much. I admire your thought. This paragraph is so bloody heavy. Okay, so this is great because you actually see the full name of these genes. To make the job of, I really find this is really stupid, to make our job as biologists as difficult as possible, the protein names and the gene names are not the same. So as very often you have in this thing, it's not the fault of the writer, it's just the way it is. But like, for example, the reverb alpha receptor is encoded by the NR1D1 gene. So I'm sort of wondering, why cannot we have the NR1D1 receptor encoded by the NR1D1 gene, right? Anyway. So now you have the definition of this ROR alpha and ROR beta. This is the retinoic acid-related orphan receptor. Orphan receptor means it's an orphan receptor. An orphan receptor means that we do not know, it looks like a receptor, but we do not know what is a receptor for. Normally, these retinoic receptor, they are the receptor of retinoic acid. But in this case, they look like retinoic acid receptor, but they do not bind retinoic acid. So we do not know what they bind. They are just orphan receptors. They may not bind anything, they just look like they could, but maybe they don't bind anything. So that's possible. There are other very famous orphan receptors, like the G protein orphan receptor. There are many receptors in the body. We know they are receptors, but we do not know what they are receptors for. They could be receptors for hormones, they could be receptors for nucleotides or whatever, but we do not know what they are. So usually the name orphan is used because we don't know what they find. Anything about these words? Don't hesitate, huh? don't hesitate. I saw nobody actually mentioned what is. Nobody asked a question about the previous paragraph, which contained all these obscure names, like for example this per and simp. Per and simp. I cannot even find back. You know about this path domain. This path domain business. Yeah, so the core circadian clock gene network about an heterodimeric partner BMAL1, which are basic helix, loop, helix, BH, LH, per, and SYNC path transcription factor. Is that okay? It's not okay, right? I thought so. Why did nobody ask? So essentially it's just a name. So these BMAL1 and per proteins, they are very big proteins and they contain different parts. Okay, they are in protein, they are called different domains. And one of the domain of these BMAL1 or per or cry proteins is called the path domain. And it's only called the path domain because the first time that this domain was found, it was in 3 proteins. The protein called per, the protein called SYMP, and the protein called ANT. So it was called path domain. And per, ANT, and SYMP, they are proteins with different functions, but they all had a domain that looked the same. And the function of this domain, of this path domain, essentially, is to interact with many things. Interact with drug, interact with protein, with whatever binding partners. It's just an interaction domain. So when you see in the future, if you are a researcher, if you see a path domain on your protein of interest, it's mainly because it could bind to something. It's a path domain that binds. Okay? So all these names are complicated, but they are actually not that important, and usually very easy to unscramble. Any question? Please don't hesitate. Don't let things.
Is the interleukin 3 nuclear factor? Is it a repressor? So when it binds to
Which one?
the nuclear factor interleukin 3 regulated. NFIL3.
Nuclear factor interleukin 3 regulated. So, yes. DBP is the activator, and MPHIL3, also known as E4BP4, is the inhibitor.
So the effect is that when in the picture, these two are drawn to be connected together. And
so we have MPHIL3 here, which is there, and we have DBP, which is there. Yes. So
when these two exist together, what will happen?
That's a good question, and actually we don't know. So what we think is that when there is more DBP than MPHIL3, there is activation. And when there is more E4BP4 than DBP, then there is inhibition. So it's only at a specific time between the expression of these MPHIL3 and DBP that you can have activation of the both mediated genes. Now if you tell me what happens when they are exactly at the same level, then it's probably you have an average level of DBP mediated gene expression. So not high, not low, just about average, possibly. Many of the full names of these genes, they actually originate from the first time these proteins or genes were discovered. So for example, this TEF, thyrotroph embryonic factors, thyrotroph, it means that this transcription factor was first described in thyrotrophs, which are specific cells in the gland, in the thyroid gland. Okay, nothing else? Okay, then we can continue.
Core clock protein interaction with chromatin and chromatin modifying complexes. At the beginning of the transcription cycle, the activators clock and BMAL1 interact with the histone acetyltransferase P300 and GRAP binding protein, respectively, to acetylate histone and provide an accessible chromatin state for transcription. Clock has been reported to have intrinsic histone acetyltransferase activity and to acetylate histone 3 on lysine 9 and lysine 14 residues. The NAD plus dependent histone deacetylase sirtuin 1 associate with clock BMAL1 and PER2 and a circadian rhythm in NAD plus level driven by the expression of the clock BMAL target gene, named in turn lead to a rhythm in SIRT1 activity that feeds back to inhibit the clock BMAL1 complex.
That's also very easy to read, isn't it? Okay, so we have a lot of new stuff there. Anything? Anything I should explain from this one? It might be a little bit. Yeah, that's mostly his own work.
And so the clock BMAL interact with the histone acetyltransferase.
Yeah. Yes.
So how does it interact with acetyltransferase?
What do you mean? How does it interact?
For example, it activates the acetyltransferase and then does.
What is acetyltransferase?
The plus acetylene to the histone protein.
Yes, they add acetyl to histone protein.
Yes, and so they add in which part of the chromatin?
Well, it's written here. This is even like, for example, clock has been reported to have an intrinsic histone acetyltransferase, histone H3 lysine 9. So this is the lysine 9 on histone 3, which is typically a mark for the transcriptional activation. So what I showed earlier, we can now complete, is that these histones have a tail that can be modified by many things. It can be modified by acetylation, it can be modified by methylation, it can be modified by phosphorylation. And this tail is quite long, and many amino acids can be modified in different ways. But each modification has a different meaning. And H3K9 is typically a marker for activity. So then what happens? In relation to what I said before, what do you think happens? When clock and BMAL bind a specific available DNA, what will happen to this thing?
It's unwind.
They will unwind, yes. So when clock and BMAL, for example, imagine this is per gene, you have clock and BMAL. that will arrive here, and because of the activity of clock, maybe it will acetylate the H3K9, and then the histone will relax, and then you can have transcriptional activation. Because these clock genes have intrinsic histone remodeling activities via acetylation of histones.
So clock and BMAL bind to some specific part of some specific chromatin at some parts, and then it will activate. It will recruit histone acetyltransferase.
Yes. Yes.
So it's not. So at which protein does clock and BMAL bind to?
Clock and BMAL, what is believed now is that clock and BMAL bind to DNA. But when it binds to DNA, it will recruit other acetyltransferase, or it may have its own acetyltransferase activity and will acetylate nearby chromatin, nearby histones, so that the local histone will make all the DNA available for gene transcription to start. So clock and BMAL will not bind protein. Well, they will bind to DNA first.
But so they bind to which part of the DNA? So,
What do you mean, which part of DNA?
for example, there are 23 chromatins, and there are many places. So how do the clock and BMAL complex know that they need to bind to that part of DNA?
We've seen that in the previous lectures. We have seen that. Do you remember anything? We have seen it today. Somebody can help her.
It's the sequence.
It's the sequence.
Oh yes. So, they just bind to the E box.
Yes. And what is the sequence of the E box? You. CSC-GTG. Yes. CSC-GTG. When you have a CSC-GTG, that's an E box, and clock BMAL will bind. But that's not all, because there are many CSC-GTG. How many times? What is the frequency of CSC-GTG? What is the frequency of CSC-GTG? If you think quickly, mathematical, quickly, 4096. So every 4096 nucleotides, you have a CSC-GTG. But there are so many CSC-GTG that are there, but they are not activated by clock BMAL. So only having a CSC-GTG is not enough. There are other things, maybe the local context of the DNA, so we don't understand everything. But usually, when you have a CSC-GTG at a good position, then it is bound by clock BMAL. Yes. All right. What else? Okay. So this NAD plus dependent histone deacetylase SIRTUIN1 associates with clock BMAL and PER2. So what's the meaning of that? I mean, he's just mentioning it like that, but what is the meaning of this sentence? So far we have this, that clock and BMAL, they function and they associate with histone acetyltransferase, blah 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 blah. But then he mentions the NAD plus dependent histone deacetylase SIRTUIN1 associates with clock BMAL and PER2. And I especially attract your attention on the NAD plus dependent histone deacetylase SIRTUIN1. What is the meaning of that? What does this? What is the consequence of having that? So it's a deacetylase, so it's something that will reverse the activation by clock BMAL. but in a NAD plus dependent manner. The NAD plus dependent. What does it mean, NAD plus dependent? What is NAD plus?
It's also used in the electron transport chain.
It has a part in the electron transport chain in the mitochondria, certainly.
So when there's glucose,
Yeah, it's linked to that. So
when there's not glucose, when there's no glucose, it is the acetylase.
So more generally, it is linked to the energetic state of the cell. NAD plus means that it has been oxidized, and this is a couple, NAD plus NADH, and NADH is regenerated by the electron transport chain, and it's a very important reducing agent in the cell. So actually it's a hydrogen atom plus an electron that can be transferred from the NADH to other molecules to reduce molecules. So it's very important for the metabolism of the cell. So that means that there is a way by which the metabolic state of the cell can regulate the clock. So let's just say that for now, that the clock regulates metabolism via regulation of gene expression, but metabolism also regulates the clock. When, for example, there is not enough energy in the cell, there is a way by which the cell can tell to the clock, oh, you have to be careful here. Let's keep this image. Okay, let's keep this image. So the clock regulates metabolism, but metabolism feeds back to the clock as well, via notably NAD plus dependent histone deacetylase.
So when there's NAD plus, the deacetylase is activated or inactivated?
Yeah, so when there is NAD plus, it's activated. And when there is a lot of NADH, then it is silent. But we will see that in more details later, about the link between metabolism and the clock. Alright, I think we have time for maybe one or two more paragraphs, unless there is something else. It should be okay. Okay, yeah, so there is something that he has not explained because it's his own work. NAMPT. What the hell is NAMPT?
NAMPT.
The NAD plus dependent histone deacetylase H-dexedrine SERT1 associates with clock BMAL and PER2 and a circadian rhythm in NAD plus levels driven by the expression of the clock BMAL target gene. NAMPT in turn leads to a rhythm in SERT activity that feeds back to inhibit the BMAL clock complex. So what do you think might be NAMPT? It's obviously his work because he didn't feel like explaining.
Something between the regulation of clock BMAL and the NAD plus?
Yes, absolutely. Absolutely. So what could it be? What could it be? NAMPT. It's obviously a name of something. What is NAD? What is the complete name of NAD? It's not just NAD. It means something. Nicotinamide.
Adenosine.
It's a nucleotide composed of adenosine. Nicotinamide, NAD, nicotinamide, adenosine. Yeah. So NAMPT. the Na of NAMT is also nicotinamide adenosine. So actually, this is the enzyme that actually produces NAD. This is the enzymes that salvages NAD. Because NAD is newly made as well. Not only it's used and transformed into NADH, but it can also be made de novo. New NAD plus can be made. And so this is the function of NAMT. So there is definitely a link there, a very strong link between metabolism and the clock. So the clock regulates NAMT, NAMT regulates the synthesis of NAD, but the availability of NAD plus will also regulate in turn the clock. So it's a very intimate relationship between the biological clock and metabolism. Okay. Next. Clock and BMAL have also been connected.
Clock and BMAL1 have also been shown to be associated with the methyltransferase MLL1 mixed lineage leukemia 1. Interestingly, MLL1 interacts with the delta 19 activation domain of clock and is thought to be important for the recruitment of clock to DNA. Moreover, MLL1 is associated with rhythmic H3K4 trimethylation H3K4ME3, a common histone marker associated with active gene promoters. In addition, MLL1 is acetylated and the metabolic regulator SIRT1 rhythmically deacetylates MLL1, thereby affecting its methyltransferase activity. The related methyltransferase MLL3 exhibits circadian occupancy and is associated with promoters bearing rhythmic H3K4ME3 marks. However, MLL3 recruitment to DNA does not depend on BMAL1 or clock.
Okay, it's much easier to say methyl. It's much easier to say methyl than methyl.
Methyl.
Yeah. So, as far as I'm concerned, this is the typical case of trying to actually cite as many papers as possible that you have published. But essentially, what this paragraph and the next mean is that there is also a link that has been found between the clock and the methylation of histone for the regulation of transcription. Okay, and methylation is a little bit more complex, I think, than acetylation. Not only there is apparently a link between both, so you can have acetylation of histone only if you have previous methylation, or you can have methylation only if you have previous acetylation. So it's a little bit more complex. But with methylation, one more complexity is that it depends how many of them. Because you can have one methyl, you can have two methyl, you can have three methyl, and depending how many methyl you have, it might have different effects on activation or inhibition. You don't need to remember that, but just remember that all these histone modification is an important part of the clock, how the clock regulates transcription by modifying histone acetylation and histone methylation, okay, in a metabolism dependent fashion. So he has explained H3K4 trimethylation is a common histone marker associated with active gene promoter. So that's one of them. And the next paragraph, which I will read, and will actually end up this methylation business, is clock and BMAL1 also interact with the lysine-specific histone demethylase JARID1A and LSD1, which are both thought to promote the recruitment of clock BMAL2 PER promoters to enhance transcription. BMAL1 interact with TRAP150, thyroid hormone receptor associated protein 150, a coactivator that promotes clock BMAL1 binding to surveillance target genes and links clock BMAL to the mediator complex subunit MED1 to recruit the general transcriptional machinery. RNAP2, RNA polymerase 2, is both recruited and initiated on a circadian basis, as discussed in more detail below. Okay, and I think this is where we stop. It's 10 minutes quick, but I think we have seen quite a lot of new things today. Mediator complex. What is the mediator complex? What is the mediator complex? Anybody knows what is the mediator complex?
Is that just the regulator which governs like the balance between?
That's interesting. Yeah. So the mediator complex in bacteria, you don't have mediator complex because you have a gene. You have the coding sequence that codes for the protein. And usually you have the promoter sequence very immediately close to the gene, so that when it recruits the RNA polymerase, and there are other factors like the sequence, whatever the details, then it binds and then it can start transcription immediately. That's in bacteria. But in eukaryotic cell, the distance, what is the promoter, is very difficult, because sometimes you have a very long bits of DNA and the coding sequence is there, but EBOX, for example, maybe CAC GTG, it may be like, I don't know, 5 kilobase upstream of the gene. That's quite a long distance, right? So what that means, that clock BMAL binds there, and how the hell can it actually activate transcription of the gene 5,000 base pair downstream? And the mediator complex is a complex of protein that can actually... The mediator complex, it's a complex of protein that can... So imagine this is the coding sequence of the gene, and you have a long promoter, but thanks to the mediator complex, we don't completely understand how it works and what is the molecular configuration. But the mediator complex is something that can make things like this in DNA. And then here you may have the EBOX CAC-GTG with the bound clock and BMAL, but the mediator complex will make sure that the RNA polymerase and many other proteins can associate locally at the right position to start transcription. And that's the mediator complex. It's a big complex that can actually bring together different regulatory influence locally to initiate transcription. That's the mediator complex. That's the mediator complex. And actually, having such a long distance between maybe the EBOX, but it's not only a clock discussion. Many things are like this. Maybe distance is actually important. If it's too close, it may not work. Okay? And then you may have the mediator complex with many different proteins that under the right condition leads to gene expression. So for example, I can imagine I told you that the genes that are rhythmic in the liver are different from the genes that are rhythmic in the brain, for example. So how does that happen? Why are the genes in the liver rhythmic? They may not even be expressed in the SCN because the liver is different from the SCN and the function of the liver is different from the SCN. So maybe this thing may happen in the liver. Maybe imagine this is a gene that is important and is expressed in the liver. Maybe there is a protein that is important for the recruitment of this mediator complex that will bring all these things together and make this gene expressed and rhythmic in the liver. But maybe this important mediator complex will not form in the brain because you have one protein that is essential for the recruitment of this complex is not expressed in the brain. And so actually this association will not happen in the brain because some of the members of this complex are not expressed. And then even though you have an E-box here, CACGTT, even it's there is no local mediator complex, so this gene will not be rhythmic. That's one of the possible explanation of why some genes are regulated by the clock in some tissues. Okay, so if you have any questions, please ask, otherwise you are free to go. So please bring this paper next week.
Isn't it Golden Week next week?
Is it not? What?
Is it? Do we have a lecture next week?
Do we?
Because isn't it Golden Week?
It's Golden Week already.
Is it? Yeah.
Oh.
We are having a lecture next week. No. It's Golden Week already. No. I was thinking having a test after Golden Week, but I won't be a pastor. No. Would you like to have a test after Golden Week? No. Okay. But we will have a test at the midterm. Let's have a test at midterm. So, as we discussed earlier, before you arrived, there was a student here who may not come anymore because the level was a little bit too high. His second year, or his only first year. He may come next year, but he was asking me what will be the test in this lecture. The test in this lecture will be a little bit unconventional, I suppose. So what it will be is, upstairs I have a bag filled with little papers, and then when you come here for the day of the test, the time at which you arrive will be determined in advance, but essentially you take a piece of paper, and on the piece of paper there will be a question. Then, if you like the question, that's okay, you go at the back of the room and you prepare your answer on a sheet of paper, and then you have 30 minutes to prepare your answer, and after that, after you have prepared your answer, then you come in front of me and we are having a discussion, and we discuss about your answer. Not only we discuss about your answer, but we will also go throughout everything we have seen so far and discuss about different things. So you may see this discussion as a scary thing, but this is the opportunity for you, even if you did not really get the question that you can answer, I will actually see how well you understand the whole thing by actually, sailing with you through the lecture. So it's more like a discussion, see how you think, see this, so that's it. The midterm will just be like a normal test.